Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Welcome to our MIT Alumni Research Slam, Course 7. My name is Joe McGonigal. I'm delighted to be here with you today. I'm the Director of Alumni Education in the MIT Alumni Association, and I'm joined today by Vivian Siegel.
I'm Vivian Siegel. I'm a lecturer in scientific communication in the MIT Department of Biology, and will be serving as your host for this debut online research slam, this one spotlighting Course 7 alumni. Before we get started with our presentations, I'd like you to meet our judges. We have Bob Pryor, who's executive editor of MIT Press, Ari Daniel, who's himself an MIT PhD alum, who's a science journalist, and last but certainly not least, Holden Thorp, who's editor-in-chief of Science and the Science family of journals. So judges, could you warm us up a bit by telling us where you're calling from, what your new normal looks like in this time of quarantine, and maybe what you're most looking forward to this afternoon?
Well, I'll start. I think mine was the first name mentioned. Good evening. Bob Pryor. I'm in Exeter, New Hampshire, where I normally live. I'm used to going into Cambridge periodically, and I haven't done that in 7 weeks. I'm looking forward to listening to some interesting talks this evening. I participated in the last one of these in real time during the AAAS meeting in Seattle in February, which now seems about 8 months ago instead of 2 months ago. Joe and I had a big crowd at the, I can't even remember the name of the bar now, but we had a very good time in Seattle, and looking forward to this evening.
Hi, everyone. I think I was next in line there. My name's Ari Daniel, and I am currently in Somerville, Massachusetts. And I'm so excited to be here. Thanks very much for the invitation, Vivian and Joe. The new normal for me looks like the fact that I'm getting to spend lots more time with my two children who are 3.5 and 1.5. So we're making lots of art, we're baking a lot, we're getting outside a lot. We built a pillow fort for the first time today. I'm most looking forward to, I've done this for a few years, and I just am always so delightedly surprised by the offerings that you guys provide. And so I'm just excited to be delighted again.
Hello, I'm Holden Thorp. I'm the editor-in-chief of the Science family of journals. Thanks to Vivian and Joe for including me in this. I've been looking forward to it. I'm coming to you from Orlando, Florida, which is where my home is. I normally would be in Washington, D.C., where I have an apartment and where the AAAS offices are and where I normally go to work. But a lot easier to do social distancing down here. And also I have 2 kids who are in higher education. One is an undergrad and she's at home going to college online. So I'm sure if you could talk to her, you could share a lot of funny stories with her about that. And my other child is a graduate student at Columbia. So like a lot of you, he's not able to go into the lab right now. And he's waiting anxiously to when he can get to New York. We are working really hard at the Science family of journals to get out a lot of the research. You are seeing that science is charging along at a very high social science and all the things that we normally see. So things have been going along just fine at a very frenetic pace. So I've been
3 minutes or less. You get 1 slide. Animation's okay. We will, 10 seconds later or so, we'll make some less subtle sounds, pitches using the following criteria. Topic in terms understandable to everyone. Your attention from the beginning of the presentation and hold it. Problem, question, or goal.
I'm at an age where no glass looking at your presentation, so don't be thrown off by that.
One question from the audience. I've just entered the names of all 9 competitors into the chat. Competitors at our brick and mortar research slam in favor of donating the equivalent of our cash prize to MIT's will be made in the winners' names to the covenant and some good tips on communicating their science. All 9 of them said, fine, no problem. I'll still show up. I still want to compete. I think that's a good testament to, business as usual for who are still wanting to improve and still do from a hat for our 9 get ready to take some notes. Hannah Starobinets. And fun fact about Hannah, let me pull it up here. Hannah's calling us today from Lexington, Mass, where she's in quarantine with her husband and her 1- and 4-year-old daughters. Let Hannah take a moment to share her screen with us. And Hannah, the floor is yours when you're ready.
Thanks. All right. Hi, everyone. I'm Hannah, and I want to cure cancer. I know you're probably rolling your eyes thinking, here we go again. But what if I told you that our bodies are curing cancer right now? It's true. Every time a cell divides, just one mistake in the wrong place could turn it into cancer. Luckily, our immune systems evolved to fight enemies both external and internal, such as cancer. Our immune cells police our bodies for these criminals and destroy them. At Genosha, I research cancer therapies that leverage the immune system. In every patient, immune police officers, antigen-presenting cells, collect evidence of cancer cells, antigens, process that evidence, and show it to their supervising immune detectives, T cells. Every T cell in the body recognizes a different piece of evidence, and when it does, it becomes activated and sends out a bunch of different signals. Some recruit more cells to the scene of the crime, and others act as deadly weapons. Usually this system works quietly in the background, and we never realize anything happened. But sometimes those cancer criminals get sneaky and find ways to put brakes on our trusty T cells. At Genosha, I design therapies to get past those brakes. Using our Atlas assay, I make a library of every mutated antigen from a patient's tumor. In my research, though, the patients are mice. Add immune cells and analyze what T cells responded to. You can see on this graph at the bottom that most of the responses fall into a middle ground, but a handful of antigens cause a high T cell response. We manufacture these stimulatory antigens into a vaccine that trains a patient's T cell response to destroy their tumor. But you might also notice something pretty weird happening at the bottom of this graph. We discovered that some antigens had the mysterious ability to turn off a T cell response. This was totally unexpected. And we definitely want to exclude these inhibitory antigens from a vaccine that's supposed to boost the immune response, not turn it off. Vaccinating mice with stimulatory vaccines can slow tumor growth, but vaccinating identical mice with inhibitory vaccines can accelerate it instead. Traditional drugs have extended many cancer patients' lives, but are rarely permanent cures. The first cancer immunotherapies took the brakes off T cells and opened a window to curing cancer. Now, with personalized vaccines, we're not just taking the brakes off the immune system, we're also giving it a steering wheel and a map. We target the right antigens and exclude the wrong ones because #targets matter. Thank you.
Thank you, Hannah. And stick around for a question here. If the audience has one, feel free to use the Q&A. First question in from Ichiro. It's, can personalized cancer vaccines cause autoimmunity?
Well, the personalized vaccines we design are against cancer mutations. So they should just be targeting a mutated protein, which is specific to the cancer. So it's not targeting just like any gene in the cancer, just the mutated ones, and your normal tissues throughout the rest of your body won't have that mutation. They'll have like the normal version of that gene.
Great, thank you. We'll stick to that one question rule, and well done, Ichiro, typing faster than everybody else. Thank you, Hannah. We don't applaud in a virtual slam, I suppose, but feel free to put lots of positive feedback in for Hannah into the chat. And you can even make a Go Hannah sign if you're cheering for her today, and take a picture of it. But thank you, Hannah. Next up is,
Before we go on, Joe.
Judges.
So judges, do you have some feedback for for Hannah about her presentation.
Hannah, this is Bob. I'm curious, you didn't say what was required in order to move from mouse model to human.
So the company is doing clinical trials and mouse research in parallel. So I do mouse research, and in parallel, we're also doing the same things on human patients.
With any specific cancers?
Actually, pretty much any solid tumor. So anything except leukemias.
And I hear a lot of explanations of cancer immunotherapy, and that was excellent. So,
Oh, thanks.
nice job.
I agree. Very nice. And Hannah, good to see you again.
Hi.
It's very hard. It's a complicated thing to explain.
Yeah, that was great. Thanks, Hannah. And the one question I had is I actually found it really useful to see drawings of data instead of the real data. But of course, it left me wondering, how does your data actually compare to those kind of sketches that you had for us? So that's the kind of flip side of cartooning something out versus actually showing what's really there.
Sure.
Good. We will ask Hannah, feel free to get into the Q&A and answer questions from your other fans. But next up is Suzanne Epstein. And Suzanne, feel free to get your slide up, and whenever you're ready, you can start. I will start the timer for you, Suzanne, once you're comfortable and start talking.
Okay, so do you want fun facts, and you want me to say where
I'm sorry, I have a fun fact for you too. I'm getting into the groove here. Suzanne, fun fact, down in the FDA in the Beltway. Very little research. She works for the Center for Biologics Evaluation and Research, CBER, and the FDA, and very little research is allowed currently, she says, but we're handling the huge influx of regulatory work from home in Maryland, where she's self-isolating with her husband and a supply of oatmeal and legumes. And Suzanne, take it away.
Thank you. I'll be talking about universal influenza vaccines that can protect against all strains. Influenza is a recurring worldwide public health problem. The virus infects both humans and animals. And like the currently circulating coronavirus, a new strain of influenza can spread throughout the world very quickly due to our dense population and travel. The envelope proteins on the outside of the virus differ from strain to strain. It's difficult to predict what strain is going to emerge next, despite a worldwide surveillance network. And the current influenza vaccines we have for seasonal flu rely upon antibodies to these envelope proteins. Therefore, they have to be strain matched and they have to be changed from year to year. An alternative is to base a vaccine on conserved proteins of the virus that do not change much from year to year, and that also add another potential mechanism of protection, cellular immunity. Our white blood cells include some termed T cells that clear viruses from the body and provide protection. My lab studies universal influenza vaccines based on these kinds of conserved proteins, and they're expressed from a viral vector in the recipient. In a typical experiment, animals are immunized, and this is intranasal, not an injection. The reason for intranasal immunization is it generates immune memory at the site of infection where it's needed in the respiratory tract. We then wait 2 weeks to more than a year. We test for antibodies in the blood and in the respiratory fluids, and we test for cellular immunity in the lungs and other sites. We then challenge the animals with diverse influenza viruses. Results show that the antibody and cellular or T cell responses appear early and last for more than a year. We find that protection also appears early and lasts for more than a year. The protection is broad, protecting against both viruses matched to the vaccine and mismatched, including highly lethal bird flu. We also have found that this type of vaccination reduces transmission to others. It reduces transmission to uninfected animals placed in the same cage. Humans have these similar forms of immunity, including T cell immunity to highly conserved antigens of influenza virus and this approach to vaccination is being tested. The public health benefits of this type of vaccine are that it could be used off the shelf when matched vaccines are not available. It can reduce illness, death, and spread to others, and it can help control unexpected outbreaks and pandemics. Thank you.
Thank you, Suzanne. Certainly timely. This is my first pandemic, and I imagine for many of our viewers. A question from the audience, please use the Q&A. You have a question for Suzanne. Jan says, are these vaccines patient-specific or tumor type specific, Suzanne?
That sounds like a question for the previous speaker. These are
I'm sorry, I'm looking at the wrong ones, Suzanne. Pardon me.
It's okay.
Any questions for Suzanne? We can start with the judges.
I really appreciated you kind of drawing the parallel to coronavirus. I think that's always an effective technique when communicating something to kind of make it relevant. I mean, the flu's already relevant, but to make it additionally relevant. I just had a quick question on what the timeline for when you think this sort of might be ready for use.
As a small lab, I'm not running the clinical trials. And I must say the approach that I've been studying for 20 years is being pioneered in the UK and in Australia. But in this country, there's been more of a focus on the hemagglutinin stem, a different conserved site. As to how long it will take, that depends upon the success of the trials. And ultimately, I would hope to combine these antigens with the HA stem. A cocktail would be the best of all.
And do you think that host-directed approaches to flu are also worthwhile as a complement to this?
Do you mean individualized?
Well, I mean, we're hearing a lot about host-directed approaches to coronavirus.
Oh, in terms of treatments or
Yeah.
modifications? Possibly, but I think for a broad-ranging public health intervention that could be used throughout the world, I think probably vaccination is more promising. Those kinds of interventions might not be as long-lasting. I mean, like IL-6 receptor intervention or...
Right. And so do you think that maybe you said this, but the acquired resistance to the universal vaccine will be greater than with a seasonal vaccine?
Oh, definitely. The seasonal vaccines are poor, even for strain matched protection. They are usually inactivated virus other than flu mist, and they are really quite poor. And this approach focuses intensely on just the antigens you need that are widely shared among all strains. It's also a more powerful form of induction of immunity by using the vector.
And are they less likely to mutate in a way that would make the vaccine less effective over time?
Yes, because these antigens have been under selective pressure throughout history, whereas the HA stem has not been. That's the reason for my preference for nucleoprotein and matrix. Those are the actual ones we're studying. The HA stem is typically overshadowed by the head group and has not been under immune pressure. These have been, and there's one report from the Netherlands of an escape mutant in one of these conserved proteins, but it did not become a pandemic or anything of the kind. And immune pressure on these sites has not led to a great diversity. Presumably these are required structures.
Right.
A question from the audience, are these vaccines patient-specific or tumor type specific? Sorry if you already answered that.
That's for the first speaker.
Yeah.
That's still the question for the
Oh, sorry, I'm so
for
sorry.
Hannah.
And Ben says, what does it take to get this into people? Is that you?
It is
too?
in people currently. Vaxitech has a clinical trial going on in the UK. What it would take in this country is some politics and funding and a committed leader. But it's in clinical trials already in other countries.
Judges, anything else?
It's hard to see support for infectious diseases going down. Let's hope.
Let's-
Anything else from the judges? Thank you. Thank you, Suzanne. We have Amy Norovich up next. And Amy, if you want to get your slide ready, calling in from New York City, where she's a postdoc at Columbia. She is currently, she says, self-isolating with her partner, who's a pianist, forced into silence against his will for the duration of this slam, as well as our 2 cats and 3 betta fish, she says, who are all refugees from her lab. I'm glad somebody thought of the betta fish. Amy, take it away.
Hi, everyone. Thank you so much for the opportunity to share my work today. At some point in your life, you may have heard the claim that men and women see the world differently. Recent studies tracking human eye movements have shown that this might actually be true, that men and women process visual cues differently in ways that have profound implications for human behavior. But what is it about the male and female brain that actually differs in terms of how visual information is processed? And how might male and female brains transform the same visual cues into different behavioral responses? This is actually a more difficult question to answer than you might think. Common lab animals like mice and flies actually don't rely much on vision. Their behavior is guided mostly by chemical cues like odors or pheromones. So to understand how males and females see the world differently, we need a new animal model with behavior that is turned on by visual cues and that differs between the sexes. This is why I'm studying a fish called betta splendens. Bettas are more commonly known as Siamese fighting fish, and they're famous for their extreme male aggression. That aggression is the result of hundreds of years of selective breeding in Southeast Asia, where betting on the outcome of betta fights is actually a very popular pastime. So when a male betta sees another male, a dynamic aggressive response is triggered in both animals. We know that beta aggression is turned on by visual cues, and it occurs when fish are in separate tanks, as is the case here in the top video, where I'm showing you 2 males interact, and even when a male sees its own reflection. During an interaction between a male and a female, which you're seeing below, the male again engages in display behavior, but the female on the right doesn't reciprocate, even though she is also the result of selective breeding strategies that increased aggression in males. These male and female responses are quantifiably distinct, and we're able to show this in an unbiased manner using machine learning methods. So what's the difference between male and female brains? How are male and female bettas able to respond so differently to the same visual cue of a displaying male? One possibility is that brain activity differs between males and females in response to a given visual cue. When neurons in the brain are active, they undergo molecular changes. I use this to identify neurons that are engaged during a social interaction by detecting these molecules in the brains of male and female fish. And here I'm showing you a small section of fish brain in the male on the left and female on the right, where each red dot is a neuron activated by the sight of a male fish. And as you can see, there are many more active neurons in males than in females after interacting with a male fish. But this brain region doesn't receive visual information directly from the eye. So how could it be involved in influencing visual perception? To find out, we can inject a chemical tracer into the active region to reveal which distant parts of the brain send and receive information to our region of interest. By connecting these dots between brain regions, we can learn how visual information is modified by brain regions that differ between males and females. So by studying the brains of these fish, we're moving closer to understanding how the sexes actually do see the world differently. Thank you.
Thank you, Amy. Stick around here for any questions from the audience. We'll take 1 question per competitor from our attendees. And Alex has the first one here. Is there a genetic manipulation you could make to increase aggression in female fish?
To increase it in female fish. So if this brain region that we're interested in is actually responsible for dictating this aggressive behavior, we could try to figure out either how it's wired or what genes are maybe differentially expressed between males and females that could actually allow us to, turn this on in females and increase their aggression. So that's definitely a possibility, but as of now, this is all very new work in a very new model organism.
Hi, this is Bob, one of the judges. Amy, I have a question about the methodology here.
Sure, yeah.
These are freely swimming fish. So how are you extracting your data?
Yeah, so this data that I've shown you is actually in fixed tissue. So you might have heard of a type of experiment where we look at things called immediate early genes. So these are this class of molecule that's turned on in response to activity at a given point in time after a stimulus has been delivered to the animal. So what I've been showing you here is actually sort of a fixed section, a moment in time, but we're actively pursuing methods to actually look at activity in real time, like calcium imaging and electrophysiological methods.
Okay, thanks. One more question, if I may, and I apologize if this sounds weird. Why? What's your hope here? What are you going to do with the information that you extract about the differences? Let's assume there is a difference.
Sure, yeah. I mean, it's actually, I think, a very understudied question how the brains of male and female animals are different. And one sort of longer term goal that I think this system is very cool to study this question in. You can actually give female fish testosterone and turn them into males, right? So you can study this sort of intermediate state where things are in flux in a way that's much easier to do than in mice, for example. So it gives us a way to study the role of hormones in terms of shaping neural circuits and shaping behavior.
And can you study this in brain development?
Yeah, we absolutely could. And it's much easier than doing in a mammal because these guys fertilize externally and they actually have a really cool courtship and mating behavior as well. So we can collect eggs, fertilized eggs at very early stages, like 2 to 4 cells. And so any manipulations that we apply would be very early on, and we're less likely to get the sort of chimera situation that you would get in a mammal.
Amy, I had a question. You set up the project by talking very generally about males and females seeing the world differently and how other model organisms might not be so good for studying this and the fish could be. Setting up that problem actually anticipates that at some point you can go back and apply what you're learning here to, some might say the proper study of man as man, like back into other organisms and potentially mammals and humans. Where do you see this work going in that regard?
That's a good question, and it always gets very tricky from a moral perspective, I think. I'm interested in this, I think right now, at least purely from a scientific perspective in terms of investigating how male and female brains may or may not be different, which I think is still a hugely open question at this point. I almost don't want to find that to be true, but the science goes where it goes. And I think it's a very interesting line of questioning to follow.
Okay.
Judges, anything else for Amy? Thank you, Amy. And again, for our viewers watching today, feel free to lobby for your favorites in the webinar chat if you wish, or follow up with them in the chat too. We have Helen Ho up next, and Helen, if you'd prepare your slide. Helen is joining us today from Manhattan. As the wildlife flourishes in Manhattan, she told me during the COVID pandemic, Helen's been waking up to the windowsill birds Philharmonic. Poetically put. Helen. And I'm seeing your slide, Helen. I'm seeing a little bit of distortion on it. Amy, are you still in SlideShare? No. Helen, are you on mute? And I can't hear you.
Can you hear me now?
Yes, there you are.
Perfect. Okay.
Great.
Thank you so much, Joe and Vivian. So how does our brain tell apart between self and non-self? So imagine someone crunching on these chips next to you, okay? So just think about how loud it is. But now think about why doesn't it bother us as much when we eat, although our mouth is right next to our ears? Our loudspeaker is awfully close to our microphone, and it's often forgotten, but our brain actually turned down the sound we make so we can actually hear others better. But how? So to crack this puzzle, I recreated this scenario in lab mice while recording the neural activity from the very first center of sound processing in the brain. I record the sound of them eating and play back to them. And sure enough, the neurons respond much stronger when the sound is passively played back to them compared to when they're actually generating sound while eating. But how is this possible? A clue of the puzzle came in when we noticed that the surrounding cells, although we're in the auditory brain structure, they don't seem to care much about sound. Instead, their activity goes up and down just as the mouth is opening and closing. So they care about the movement that generates sound and then inhibit the sound output. So we then hypothesize that the brain is using the movement information while we eat to predict what we sound like and cancel that out from what we hear. And we're right now building a model to see how through the countless eating you have done in your life that your brain learns this relationship between movement and their sounds. It's more complicated than that, actually. We actually make such a rich repertoire of sounds. Eating chips sounds very different from eating strawberries. The sound of our breathing, our talking, the list goes on. How does this process generalize all the rich sounds? So we paired up with electrical engineers to record the soundscape. So all the sounds that the animal makes as heard from the animal's own perspective. And we hope to understand how the brain learns this rich relationship, this rich association between movement and sound. So why do I care so much about chewing sound? The brain region we study that mixes movement and sound is actually implicated in tinnitus, the ringing of the ear. And we hope to understand this process better to lead to better treatment. The neural principles we learn here will also hopefully inspire engineers to build smarter noise-canceling headphones. And finally, I think we're just tapping into the secret of how the brain learns the consequence of our own action, so we can differentiate self from others. Thank you.
Thank you, Helen. I think this question's for you. It's, do you think that humans use similar neural processes to screen out noises unrelated to eating? Ichiro says.
Yes, that's a good question. So the brain region we're studying, the dorsal-cochlear nucleus, has this homologue among all mammals, including human. So based on neuroanatomy studies, we believe a lot of these processes are conserved in mammals, including human. And a lot of tinnitus research has been done on this region. That said, we are tapping into a complex problem. We think that occurs in many stages in the brain. And we try to reduce this problem by studying the very first center of sound. So we think this process also can happen in other areas of the brain, in humans and in other animals.
That was great, Helen. I was curious about how the electrical engineers actually recorded that soundscape, and then once they did, what it sounded like. Was it something that sounded familiar or weird?
It sounded weird. So first of all, there are two challenges. One is to build these very miniature headphones, microphones essentially, so it can be put on the head of the mouse without influencing or the mouse's behavior, right? So they can go about their natural behaviors as they do while hearing the sound. And the other thing is, we have to be very careful about the type of microphone that we choose, right? And there's a lot of innovation there. So we're actually recording sound, not say vibration of the bone, etc.
Hi, Helen. I actually love that you used an audio prop when you started your talk. That was actually really useful. One of the things that it got me thinking about as you were talking about filtering out sound is actually this other move that's happening like across humans in terms of mindfulness, like actually starting to pay attention to those sorts of things we've learned by being in urban environments or whatever, to tune out, to actually pay attention to them and the effects of that. So I was puzzling, and I wonder if you could speak to kind of how you can tease out not just like how the brain on its surface like hears or filters out sound automatically, but how practices might alter kind of what you pay attention to and what you don't, because you do have some, at least humans have some willpower when it comes to where they direct their attention.
Absolutely. So we are so far tackling this complex problem by just focusing, you're right, the acoustic, the very first stage of it. But like what you said, the sound has many emotional, cognitive component to it, and so effective attention, a lot of higher order controls. And you know, when I ask my friends that I study chewing sounds, and half of them hate the sounds, half of them love the sounds, like what you said, for mindfulness and for breathing, etc. So I think yes, we are. My research so far is focusing on how acoustically the brain is processing this, but there's wide room to add the emotional and cognitive components.
Thanks, Ellen. Judges, any other questions or feedback about the presentation?
Thank you, Helen. And I will note that I am, Vivian, I feel like a meanie. I'm dismissing all of these great questions from attendees, but would make it known that we are going to forward all these questions onto competitors with contact info if applicable. And you can also feel free to message competitors in the chat one-on-one if you have that power, and I think you do. Next up is Maya J. And Maya, if you'd get your slide ready.
Hey everyone, can you guys hear me okay?
I can, yep. Maya says she's been making a point to run by the MIT Bio building every day during her socially distanced jogs. I'm glad, I'm glad for that, Maya. I haven't seen MIT campus in 7 weeks, so I miss it.
It's doing very well. The flowers are out, so that's great. Okay.
Good.
All right, you can see my slide okay? Great. You know, when I started learning how to dance, I had 2 left feet. It took me a lot of trial and error to perform my movements correctly and stop repeating my mistakes. And I bet many of you have probably had similar ordeals in learning skills like how to swim or how to ride a bike. Learning at its core is driven by neurons in our brains, specifically by neurons that release a chemical called dopamine. For example, I was able to learn a dance move because in the moment that I finally got that sequence of motion right, a rush of dopamine was released all over my brain. And this dopamine sent positive signals to billions of neurons to make me feel good, and thus helps teach me that performing and repeating that motion in that particular and correct way was a good thing. The release of dopamine in the brain is like a confirmation that we're doing something right. But what if I told you that when you learn to perform some action or skill, you're not just honing that one specific skill, you're actually honing multiple actions that you already know how to do. I myself felt this while I was training as a dancer. As I improved in my dance, I found that my walking gait had changed, my posture when I sat down for class had improved. So is it possible that the learning of specific actions can have general effects on several other actions that an animal is able to perform? So I am currently trying to discover answers to this question through the lens of mouse behavior. I start by choosing a particular action for my mice to learn well, such as running or grooming their fur. And then I can record videos of my mice behaving and use machine vision in real time to detect when my mice perform that specific chosen action. And whenever they do that action, I can give them that fast dose of dopamine. This real-time pairing of actions and dopamine, much like how I learned to dance correctly, enables mice to learn that specific action. But what's fascinating are the changes in all the other behaviors that I didn't explicitly target. For example, I noticed that when animals are instructed to learn fast behaviors, such as running, all of their other behaviors, such as grooming or rearing, start to speed up. And conversely, we observe a slowing down effect on those same actions when animals are taught to stay in place, suggesting that one way that learning a particular action may broadcast general effects is via tuning the speed of other actions. So maybe I can convince you that perhaps our brains are picking up principles that are far more abstract than the specific skills that we set out to learn. for the first time, we can break the classical neuroscience mold that uses behavior to study learning, and instead leverage the powers of an animal's innate learning to understand something global about behavior. And hopefully we can unlock the rules by which our behavioral repertoires are flexible, malleable, and constantly adapting to everything that you or I could ever learn how to do. Thank you.
Thank you, Maya. Stick around for a question from the audience while the judges tabulate. Any questions for Maya on this? Please enter them into the Q&A feature if you haven't already. And judges, I'll say while we're waiting to please chime in at your leisure.
Yeah, that was
So-
great. Oh, go ahead.
Go ahead, Ari.
No, please go ahead, Holden.
So is there I realize, you would only have a guess at this, but is there a logical way to imagine the mechanism of this? Are there structures in the brain that would be influenced by learning a fast behavior that would then be utilized later for something else?
That's a great question. So understanding how actions are actually encoded in the brain is a really, really tough problem. There's a lot of different motor centers. They're all passing information to each other in kind of redundant ways. And the problem is that dopamine is dumped onto a lot of these centers at the same time. So it's kind of hard to disentangle exactly how dopamine might be shaping behaviors on a specific and a general way. Our general theories of how learning by reinforcement works via dopamine sort of has this sort of action and then reward. And then every time you get rewarded by performing an action, you can do more of that exact action. And in the brain, this has sort of been posited by, oh, well, there's maybe a certain circuit of neurons that is, has some activation program. And if dopamine is dumped onto those neurons, when that program is active, those neurons now undergo plasticity, or they just become more likely to become activated. So you end up doing that action more. That's sort of the canonical model of how we think reinforcement works. But realistically, dopamine is being dumped on a lot more than one particular action program worth of circuits and neurons at once. So likely what's happening is that there's multiple groups of neurons, multiple ensembles that are maybe encoding more abstract things than specific action programs. And dopamine is maybe tuning all of them, making all of them more likely. So it's the same reason that I might be more likely to sit up better and run faster when I learn how to dance faster or something like that. So you can think of ways that maybe the things that our neurons are encoding is not like a specific action program, but maybe something that's slightly more general, and then dopamine kind of just tunes all of it in a certain direction. But again, it's just a guess.
That was great, Maya. Thank you. I really enjoyed that. I may have missed this, but how do you give the mice the dopamine?
That's a great question. Yeah, I didn't explicitly talk about it, but we use a technique called optogenetics. So opto is the Latin root that means light. And so basically we have specially engineered proteins that are light sensitive. We can express them in neurons that contain dopamine. And then we basically stick a literal fiber optic into the brain just above those cells and shine light onto these neurons. And that way we have very precise temporal control. So as soon as they do an action, we can immediately follow it with like a pulse of light. And that will activate dopamine neurons immediately after an action is performed. So that's how we do it.
Fascinating. Thank you.
My question from the audience, are there certain actions that are resistant to improvement in tandem with the action that the mice are taught to improve?
Sorry, can you repeat the question?
Are there certain actions that are resistant to improvement in tandem with the action that the mice are taught to improve?
That's a great question. It's definitely something that we have the data to look at. This is still a very much ongoing project. We do find that different behaviors that we might try to teach the animal are learned to different degrees. So they can learn some behaviors, maybe like running, more than they can learn how to like turn to the left. Those are just random examples. And so, yeah, and because they learn different behaviors to different degrees, we also see sort of a great amount of variability in the sort of neighborhoods of other behaviors that they shape. But we haven't really gotten conclusive answers on exactly that, but we are working to understand it right now.
Hi, Maya. I had a question, a kind of more general question. So those of us who've kind of watched the dopamine and primary reward literature / newspaper articles tend to think about dopamine as the mediator of addiction. addiction to sugar or food or drugs or whatever. So it's sort of interesting to see you bring up dopamine as an aspect of learning. So my question is kind of general, like having said that, would you say that our ability to learn is very much shaped by the reward that is offered at the end of that learning? Are there, I think as an educator, kind of thinking from extrinsic to intrinsic motivations, it's kind of, getting, offering someone that hit of an A grade or something like that as a motivator to get them to learn a pathway. When you take the grades away, they lose that external motivator. So I'd love to hear you talk more generally about dopamine as a learning.
Sure. Yeah. So I think I don't know if there's actually that's ever done like a direct comparison of different types of rewards and how an animal or a human might value them differently. But at least in the mouse literature or monkey or animal literature, when we study behavioral neuroscience, people have tried things from the scale of like water and food. Then there's like the whole optogenetic kind of abstract hit of dopamine. And then on the other end, there's like drugs of abuse, such as cocaine, which is known to be extremely addictive. And, rats will press levers obsessively for hours on end. So there's definitely a varying degree. And I think what's likely happening is that the reason why there are different degrees and why different rewards might be influencing dopaminergic circuitry and influencing the way downstream neurons can change our behavior is just because each of these stimuli or these rewards acts on different parallel circuits in the brain. So you might imagine that a food reward or a water reward is acting on centers that involve dopamine, but also involve, centers that involve hunger, that, have to do with hunger and thirst and like deep, like set, like satiety signals. Whereas we know that cocaine acts in, certain parts of the brain, such as the nucleus accumbens. This is a region of the brain that's very involved in valuation and is very susceptible to developing maladaptive behaviors when it receives a ton of dopamine. So my guess is that there's some sort of spatial organization to the types of, to basically where each of these rewards is kind of like hitting hardest, if that makes sense.
Thanks, Maya. Judges, any other feedback on the presentation? Anything else you want to say?
I just wanted to say I appreciate the passion. I appreciate the presentation. It was very interesting.
Thank you.
The motivation is obvious there on your part.
Thank you, Maya. Judges, we had promised you a bio break, no pun intended, after 5 competitors. Please feel free to take one if you need one, or catch-up break for catching up on your notes if you need one. But maybe I'm feeling a good pace right now. Judges, if there are no objections, I will proceed. Our competitors are being very true to their 3-minute time limit today. And audience, if you're not aware, this is incredibly hard to do for a graduate student or a postdoc or junior faculty to distill all this research in 3 minutes. Please send them lots of love in the chat if you appreciate their work. We have 4 competitors left, and we'll move to Lori Huberman. Lori, if you'd set up your slide. And Lori is joining us from Berkeley, California today, where she is self-isolating with her husband, her dog, and the family of deer that sleep on their roof. Lori, I'd love to hear all about that, but I want to hear your research first. So Lori, when you're ready, you can start.
Is my screen sharing?
It is, yep.
Okay. Let's imagine I told you there was an alien invasion of organisms capable of making cellular networks that could span hundreds of miles, was killing 1 million people a year, decimating frog, salamander, and bat species, destroying 30% of our crops, and had made itself integral to nutrient cycling in our environment. I'm guessing you'd be pretty interested in understanding the biology of these organisms. Well, it's not an alien invasion, but fungi are capable of doing all of these things, and we know very little about them. We don't know much about what they eat, where they grow, how they reproduce, or what most of the genes in their genomes do. To rapidly characterize the role of fungal genes, we're developing tools to do massively parallel screens to get from phenotype to genotype. Many of you are probably familiar with the biology of single cellular fungal organisms, such as baking and brewing yeast, which have been making themselves particularly useful over the past couple of weeks. But most fungal organisms are multicellular and filamentous. I'm showing a video here of a single filamentous fungal cell. And these fluorescent dots that you see zooming around are actually nuclei. As you can imagine, this sort of cellular morphology makes doing high throughput work extremely challenging. We're using insertional mutagenesis to make barcoded mutant libraries with hundreds of thousands to millions of mutants to do pooled mutant screens. We insert these DNA barcodes randomly throughout the genome and then use high throughput sequencing to identify the location of each of the DNA barcodes. So for instance, barcode 1 might be disrupting gene A, and barcode 2 might be disrupting gene B. Once we've identified the location of each of the barcodes, we can then expose this mutant library to different experimental conditions and then use the abundance of each barcode as a proxy for the relative growth speed of each mutant strain. So here's an example of a mutant library. Each of these orange cells, each of these orange circles indicates a cell, and the smaller colored circles inside indicate individual mutant nuclei. We expose our mutant library to an experimental condition. As most of you are sadly and yet intimately aware, most mutants have no phenotype under most conditions. And so most of our strains are going to grow at a fairly similar rate in most conditions. But some mutants, like this yellow mutant here, won't be able to grow at all. And so the barcode associated with this mutant will be underrepresented as compared to the population as a whole. And we can hypothesize that the gene that this barcode is disrupting is required for growth under this condition. Meanwhile, other mutants, like this blue mutant here, will be able to grow more quickly than the rest of the population. And so the barcode associated with this mutant will be overrepresented as compared to the population as a whole. And we can hypothesize that the gene that's disrupted by this barcode is detrimental for growth under this condition. By exposing our mutant library to a large number of different experimental conditions, we can assign roles to different fungal genes, which will help us to identify cures for fungal diseases of plants and animals, and in further developing filamentous fungi for use in biotechnology going forward.
Thank you, Lori. Questions for Lori from the audience, feel free to use the Q&A feature if you've got a question on her research. And judges, feel free to jump in when you wish.
That was great, Lori. I love fungi. I'm curious to know how you kind of opened by setting that kind of alien scenario, which was terrific. And I'm wondering, how did those like various ways in which fungi are strange and creating trouble, how does that map onto what you were just describing about in terms of growth?
Yeah, so basically, we know that fungi are able to replicate inside plants or inside animals, which is what causes sort of these fungal diseases when they're like utilizing the resources, and they have to get their food from somewhere. So some fungi get that from dead plant material or something, but some fungi are actually invading humans or plants and getting that material from live organisms. And so by doing experiments where we can change the growth speed under different nutrient conditions, or like inside a plant or inside an animal, then we can understand what genes are involved in the invasion of that organism and help to identify what genes, like what the genetic mechanism is of disease.
Great, thank you.
Steve asks, why do mushrooms get invited to all the best parties?
Because they're fun guys.
I have worked a lot on fungi single cell, and I know embarrassingly little about these. Do they have about the same number of genes?
So most filamentous fungi have significantly larger genomes. The smallest ones are around 10,000. So Saccharomyces cerevisiae, which is sort of the workhorse single cellular organism, has about 6,000 genes in the genome. Sort of the simplest filamentous species have more like 10,000, and many have upward like 15 to 30 to 40,000 genes.
Hey, Laura, I just wanted to offer a little bit of feedback on your presentation a bit. So like one of the things I noticed is you were standing up when you were speaking, which I think is awesome. Like it gave you a lot of energy and that came through entirely. I also have to admit I got a little lost listening to you. And so I just wanted to offer like one of the, you speak really fast and with a lot of energy, which is wonderful. And one thing that might be useful to learn is when you want to pause. So pauses are super effective because everyone stops and pays attention to you when you take a break. And you can really get people to what it is that's the most important thing you want them to hear by taking a breath before you tell them what it is.
Okay, thank you.
I also appreciated that you were standing up. I'm Italian, I speak with my hands, so I need to move when I'm talking as well. So I appreciated that. You, right at the very end, you gave one sentence, again, very rapidly, about where you see this going, where this research is going to be applied. And I completely missed that. Could you fill that last bit in for me?
Sure, yeah. So basically we can, because these libraries are pretty versatile, we can expose them to a bunch of different conditions. So we could expose them to different nutrient conditions, such as nutrient conditions that are associated with, such as infection, either in plants or animals, to see what genes are involved in sensing or utilizing different nutrients. We could also expose them to different conditions where we have multiple microbes at a time. So we know that microbes interact and signal to each other, potentially doing like secreting different chemicals that are actually sort of like defense mechanisms. So we could identify the genes that are involved in either sending out those like different mycotoxins or in responding to different toxins from other organisms such as bacteria. Or we could identify genes that are involved in invasion of either plant or animals. So the genes that are involved in adhering to different plants by exposing them to an infection condition.
Thank you.
Lori, a real question from the audience. Phenotype Citro says, you described at the outset seem very difficult to capture in high throughput screens. Are you missing the most interesting associations between phenotype and genotype?
So it is difficult to deal with these organisms in high throughput screens. That's like one of the big things we have to work on is how to actually get these organisms up and running. One of the great things about this experiment, because we're not looking for like a colony to form, we're actually just looking at the relative growth rate of organisms, is we don't have to let the experiment run for very long. It only has to go for maybe 10 to 14 generations. And so before we get these huge networks, you know, that would, so normally a fungal colony would just like spread over as much space as you get it. But before we let it get to the point where it's taken over, we can, you know, kill it and harvest the DNA and figure out, get the relative abundance of barcodes. And so that is a great thing about this particular way of looking at gene function.
Your figure is an excellent, and the way you described it, an excellent way to show the experiment. I think a lot of people would understand precisely what you're doing. So that was great.
Thank you.
Judges, anything else? Thank you, Lori. Next up is Julie O. Julie, if you get yours ready. Julie says, still lives in Cambridge ever since she came to MIT as a freshman and still loves living here and is calling in from home today in Cambridge.
Hi, can everyone hear me?
Yep.
Sure. Okay, so for most of human history, we have stood helpless in the war against cancer. But in 2010, the first immunotherapy drug has truly revolutionized the way we approach some cancers. The idea was that if we can detect cancer cells and train and stimulate our own immune cells to go for those cells, we improve survival. Patients with more than a dozen types of cancer can now receive FDA-approved immunotherapies. And from 2016 to 17, we saw the largest single year drop in the US cancer death rate ever recorded. However, around 60 to 80% of patients still don't respond to immunotherapy, and it is challenging to predict the success or failure of the treatment because we don't understand precisely why some patients show complete tumor regression while some patients don't respond at all. The immune response against tumor is like a tug of war between anti-tumor immune cells versus pro-tumor immune cells that infiltrate into the tumor tissue. The progression or eradication of tumor is a collective outcome of the interaction among these diverse immune players, which also changes with time. So in order to understand the drug response, it is critical to be able to analyze all the important immune cell populations at multiple time points of treatment. In clinical settings, however, it is oftentimes limited. The number of biomarkers that can be analyzed per specimen is limited due to insufficient amount of the specimen and limited resources. To overcome this problem, the goal of my research is to come up with a better way to analyze these diverse tumor infiltrating immune cells. We have developed an antibody-based imaging technology that allows deep profiling of immune cells in a scalable fashion. So this approach has advantages in multiple aspects. First, it enables analysis of all specific markers necessary to identify distinct immune cell population in a single specimen. Second, it works with a small amount of sample as they are analyzed at the single cell level with minimal sample loss. Third, it uses well-tolerated, minimally invasive sample collection procedure, which allows time course analysis of each patient. And lastly, the whole pipeline from sample collection to data analysis is done in hours as compared to currently available methods, which usually takes from days to weeks at a much higher cost. Our deep immune profiling has recapitulated surgical approaches and identified patients whose immune signatures are linked to favorable clinical outcomes of immunotherapy. Moving forward, I hope this approach can serve as a clinical tool for initiating and monitoring the efficacy of immunotherapy and contribute to many of future breakthroughs of immunotherapy in cancer immunology. Thank you for listening.
Thank you. That's Julie O. Questions from the audience for Julie? Please use the Q&A feature. And judges, please chime in when you want as I get my phone to stop chiming.
I'll just say, oh, please go ahead, sorry.
Go ahead, Ari.
I just wanted to say I really liked your visual. I thought the part up at the top, that's just such a great way to encapsulate an idea, you know, with the cells underfoot and the cells as heads. I just thought that was a really nice way to depict it.
Thank you.
Yeah, and I was going to say the same thing. Immunologists, I kid with all the ones that I work with, and over the years that they have so many different kinds of cells that, you know, they just do that on purpose so the rest of us can't really follow what they're doing. And you did an excellent job just with this figure of kind of taking that off the table so that people don't have to remember, oh wait, is she okay, a dendritic cell? That's natural. That's not the same thing as natural killer or whatever, you know. So that's always a challenge with immunology, and I think you did a really nice job with that. My question is, how much of this profile is in the germline, and how much of it is influenced by the cancer itself or by other environmental factors?
So the specimen we are analyzing is a subset of cells that we obtain from injecting fine needles into the tumor tissue. So we are targeting the tumor microenvironment to have a sense how many of these cells are there in composition of the tumor microenvironment.
Right, and so the profile of the different population of the immune cells is somatic to the tumor, right? Or yeah. So in other words, it's going to be a different result for every patient.
Yes, so we expected, for example, we call tumors that are infiltrated with a lot of T cells hot tumors, so that that patient is expected to respond to the immunotherapy much better than patients with cold tumor, where the tumor's low number of T cells infiltrating. So we think that it's like a host versus non-host response. So the immune cells are hosts attacking these non-host tumor cells. And we believe there's different immune response coming from all these different immune populations forming inside the tumor, resulting in different clinical outcome after immunotherapy treatment.
Great, thank you.
Yep.
Julie, I'm wondering if this type of technique has application other than in cancer.
Yes, so we are currently focusing on cancer, but we can definitely try this to profile any kind of environment where there are a lot of heterogeneous cell population reside and interact with each other. As long as we know the markers to target, we can always profile for all these different kinds of cell populations. Yeah.
I agree also. I think it was with Holden or Ari who said the quality of your graphic is very good,
Thank
especially
you.
the top part, the tension there.
Judges, anything else for Julie? Well, thank you, Julie. And she'll certainly, you're welcome to send her any other follow-up questions in the chat. We've got two competitors left. Alessandra Hillis, if you can ready your slide. And I think I mentioned this in the chat, but audience, stick around for a audience poll on the coolest wow factor science at the end of our presentations today while the judges deliberate. Before they name their winners, we want to hear what was the coolest wow science you saw today. So Alessandra's got your slide up, and whenever you're ready, you can begin.
Okay, great.
Sorry, Alessandra, I have a fun fact on Alessandra
Oh.
here, too, who says she is self-isolating with your family and your 18-year-old cat. Go ahead.
Okay. All of the cells in our body rely on signals that tell them what to do. Should I grow, die, eat this, or break down that? These signals come in the form of small molecules, and they activate signaling pathways in the cell. Normal cells are carefully regulated by these signals. They detect the signals on their surface and then activate the proper downstream signaling pathway. When the signal is gone, the cell stops activating this pathway. Cancer cells, however, can acquire mutations in these signaling pathways, which make them constitutively active, even in the absence of outside signals. A major growth-promoting signaling pathway that's hyperactivated in almost all human cancer types is the PI3 kinase AKT pathway. The PI3K AKT pathway is the most frequently mutated oncogenic signaling pathway in breast cancer, so drugs have been developed that inhibit this pathway. By inhibiting this growth-promoting pathway, the drugs tell cancer cells to stop growing and die. However, the drugs have some limitations. First, they have adverse on-target side effects at the therapeutic doses, which limits their efficacy. And second, cancer cells are really smart, and they'll do pretty much anything to stay alive, so after getting hit with one of these drugs, cancer cells can adapt and become resistant to the drug. My research aims to overcome the challenges of these drugs to effectively treat triple-negative breast cancer, or TNBC. TNBC is the most aggressive subtype of breast cancer, and the standard of care remains chemotherapy despite advances in treatment options for other breast cancer subtypes. To utilize PI3K AKT inhibitors effectively in TNBC, my project aims to identify combination therapies with PI3K AKT inhibitors. This would allow us to use lower doses of the inhibitors and to target the cancer in more than one way, which could potentially limit the development of resistance. To do this, I performed a CRISPR dropout screen. So let me walk you through this scenario to explain how a CRISPR dropout screen works. Suppose 4 people go to the beach, and one person has an umbrella, a hat, and sunscreen. From each of the other 3 people, I take away one of these items. In the shade, they're all fine. However, when they get hit with the sun, the people without an umbrella, a hat, or sunscreen will be more likely to get a sunburn. This combination of sunlight and loss of one of these items leads to a sunburn. We can think of a CRISPR dropout screen in the same way. CRISPR technology allows me to genetically manipulate breast cancer cells such that each cell in my population has one gene in the genome knocked out. When these cells are hit with a PI3K AKT inhibitor, some cells will die, just like some of our friends at the beach got a sunburn. This combination of loss of a gene and PI3K AKT inhibition leads to cancer cell death. Identifying these genes whose loss or dropout causes cancer cell death upon exposure to PI3K AKT inhibitors could highlight potential targets for combination therapy with PI3K AKT inhibitors in triple-negative breast cancer. The ultimate goal of this work is to characterize the first targeted therapy strategy for TNBC and hopefully one day make an impact on patient lives in the clinic. Thank you.
Thank you, Alessandra. Questions for Alessandra on her science? Please use the Q&A feature in Zoom, and judges, you're welcome to chime in whenever.
So what kind of genes do you see popping up in your assay?
So it's actually quite interesting. I've described it a little bit simplistically, but I've done this with both a PI3K isoform specific inhibitor and a pan-AKT inhibitor. And in both of these cases, different genes came out. And what's interesting for both of them is that a lot of them are related to different metabolic programs in the cell. And so I think that manipulating or inhibiting the PI3 kinase pathway potentially shifts the metabolic programming in these cells. And then by inhibiting that metabolic program that occurs when the cells experience inhibition of the PI3 kinase pathway, we're then hitting that second target, that adaptation to the inhibition with the PI3K AKT inhibitor. So there's a lot of metabolic programs that are affected.
Yeah, and so a lot of kinases change.
Yeah, in addition, there's a lot of MAP kinase, ERK, those are the things we expected from the outset. However, those are also potentially some of the most toxic combinations that we could give patients. So those are some of the things that are great validation for us, but we're less interested in pursuing for future research.
I just wanted to compliment you on your graphic. I was a little nervous when you only had the top part visible because I wasn't following you particularly well. But the use of the sunburn metaphor at the bottom is very effective.
Thank you.
Judges or audience, anything else?
I had a question. So you set up the CRISPR dropout screen very well. I love the metaphor of the sunburn. That's great. One thing that came to mind as you were talking is that you could also do this much more directly by looking at other small molecules that already exist that together with PI3 kinase AKT inhibitors actually lead to cancer cell death when they might otherwise not. Can you compare those two approaches for me briefly and talk about why you'd want to do this over that?
Yeah, so it was definitely a consideration. There are a lot of libraries of small molecules. You can take your pick, druggable libraries, undruggable, and it would be a similar approach here. The reason we chose to do this whole genome targeting CRISPR library was because we really wanted to open up to any possibility of a gene that we could identify. And ultimately, we are potentially interested in pursuing some things that could be considered, quote, undruggable. But now with the development of all different kinds of inhibitors and degraders today may potentially be druggable. So that was kind of our motivation. But it's definitely possible that using the inhibitors potentially could have taken us faster to a potential clinical outcome. But I'm in my second year of graduate school. We have plenty of time to go, so we'll see where this takes us.
Good luck on the project.
Thank you.
Question from the audience: what fraction of CRISPR knockouts are unable to survive at all, Alessandra?
Yes, so it's hard to, I don't know if I can give you a number off the top of my head, but there definitely are a significant number. You take cells at a couple of time points here, and so there are a significant number that do not survive immediately upon knockout, and that's relatively proportional to the number of essential genes in our genome. because obviously when you lose an essential gene, the cell then dies. And so we control for that in the experiment.
Thanks. Anything else from the judges? Alessandra? Thank you. And we have one competitor left. It's Allegra Hawkins. Allegra says she is calling us from New York today and has been competing in synchronized figure skating for the past 20 years. Says she's when she's not in the lab, she's at the ice rink skating and coaching. Allegra, the floor is yours.
Can you see my screen?
Yes, I can. Not full screen.
Yeah, there you go.
There we go.
Okay. Does anyone remember before we had Google Maps, before we could just Google the name of the place we were headed, and that magical voice in maps then tells you exactly where you go and even when to turn? Maybe some of you have memories like me of being in the back seat of the car and your parents folding this huge map and arguing over the best way to get from point A to point B, and then struggling even more with folding it back together. I'm sure many of you think that these maps are old and useless. But what if I told you that to diagnose many types of cancer, doctors actually biopsy tumors to create a tumor snapshot, similar to old school maps. But all it really tells you is where the cells are located. By eye, all the cells may appear to be the same, but they're actually all a little different. Some of them may carry what's called a mutation, or a change in their genetic makeup that alters their function. So some of our cells may be houses, while some of them may be local businesses. In addition, each cell or location on the map has a list of details associated with it. What hours is the business open? How many stars does it have? What are the reviews like? All of this information is similar to the list of genes expressed in a cell, and it helps determine what type of function the cell has. But we wouldn't know all of this from our old paper map. Right now, to find out detailed information about each cell, we'd actually have to dissociate all the cells in the tumor and then sequence them. That's like knowing all the places on the map and all of their details with no idea how to find them. Or like someone telling you about a store that has endless amounts of toilet paper during COVID, but not actually knowing the address. Pretty disappointing, huh? But what if we knew both the cell's location and the cell function? This is exactly the type of technology we're developing in the lab. A Google Maps for tumors, a magical search engine where we can look up the cell address, the cell type, the mutation status, the gene expression profile, and even get a link to a website containing all of its detailed information. So why do we even need a Google Maps for tumors? Again, let's go back to our toilet paper store analogy. And let's say you know the store hours and the address, but the store said only the first 200 customers will be able to buy toilet paper. So you need to get there ASAP. You need directions and a route with minimal traffic, another great feature of Google Maps. That magical voice that talks to you and tells you exactly when to turn and even when to avoid traffic. Now, the way that tumors can grow is by cells talking to each other, and the way they do this is through sending signals to each other. So if we want to stop tumor growth, we need to learn the local cell neighborhood and how these cells are actually communicating with their neighbors. This is why we need a Google map for our tumor. We need to understand the roads and the traffic patterns, what types of cells are communicating with others, and what roads are they taking, so we can close these roads, block the communication, and kill the cancer. By applying single cell analysis to our old school tumor maps, we can create a Google Maps for cancer, and soon we'll have our new advanced technology database, and maybe that little magical voice will be telling us the step-by-step directions on how to win the war on cancer. Thanks.
Thank you. That was Allegra. Please ask questions of her in the Q&A feature.
Allegra, this is Bob. I'm curious, is this useful in a clinical setting?
Yes, absolutely. So we would be able to basically take our tumor from the patient, so any sort of biopsy tissue, and then process the tissue immediately. So you freeze it directly and then take slices of the tissue and then do the sequencing from there. And so you get the information from the actual tissue like location itself, and then overlay the sequencing information on top of that.
But if the goal would be then to use a therapy to reduce a tumor rather than to aid a surgeon in removing it.
Yes, exactly. The idea is to say, okay, these are what, more of a personalized medicine approach. So if you are having surgery to remove your tumor or even a biopsy per se, then you could say, this is what's being expressed in this specific tumor in this specific patient. And can we design and pick a therapy approach based on what we're finding using this approach?
Thank you.
Dennis asks, what is the enabling technology that allows you to gather both type and location information in order to map it?
Yeah, so it's called spatial transcriptomics. And so that's the commercialized platform that's available. And then we're also modifying it by using our genotyping of transcriptomes method that we've developed in our lab. And so basically what that does is it takes a tumor, takes slices of it, and then overlays that onto a slide that has a small barcoded array. So each dot on the slide has a different barcode that's mapped to it, so that you can actually sequence the RNA or sequence the tumor and map it back to that spot that you had on the slide. And then we can also then genotype every piece of tumor that we get. And so combining all that together, you get both pieces of information.
Allegra, I thought your metaphor worked really well throughout. I really appreciated how you picked it up at the beginning and then followed it all the way through and continued to draw connections to it. It made your presentation very easy to follow. And I also love the images, especially in the lower left, the kind of all pink and then the colorful cells. I thought that was really pretty to look at, in addition to just being a nice way to represent what you're talking about.
It is kind of similar it's pretty similar to what you get from the actual data. I just wanted to make it hopefully appealing to the general public so that they're not.
You did that.
Yeah, I wanted to add on to that comment. I did find the metaphor at one level really useful, but I also found myself struggling with do I really feel this is like Google Maps? How isn't this like Google Maps? And I got a little bit caught up in that over following what you were doing. But I do think there are two threads that you brought up that are both interesting and worth disentangling if you think about doing them in some ways. One is the richness of information that exists, right? And that your slide shows super well, how much more information you get. And so when you use Google Maps sitting at your desktop, not actually trying to get somewhere, you can use all the information that's underlying it to find things out about the place you're going and so on. And then there's that other piece you keep bringing up about the step-by-step guidance you might get without ever even looking at the map.
Right.
And that's where I think what I would love to see if you gave this presentation again is a way to talk about the richness first and then go to that step-by-step thing separate, because I think that's in some sense the future, what this could enable and how it does it in Google Maps might be helpful to disentangle those a little bit, at least for me. Yeah.
Absolutely. It's a complicated analogy. There's a lot of information in Google Maps, I think we don't even realize. So
yeah, and the other piece I started asking myself when I was thinking about it was does each individual person have their own private Google map? Or are you doing predictive Google Maps based on a population or someone else with a similar genetic makeup, that it's sort of predictive because rush hour yesterday was like this? We don't really know the actual traffic patterns in your tumor right now, but we could guess at them based on all this other information. Those are two different ways this metaphor might be playing out. Both are possible, right? So
yeah, absolutely.
Any other comments from the judges? Presentation, science?
Good. Well, we will take a 10-minute break to allow the judges to sequester themselves in a private Zoom room chamber under lock and key. Judges, off with you.
We're going to leave this meeting for a little while.
When we see your faces again, we'll know your decisions have been rendered and you'll announce them. And in the meantime, audience, I'd love to hear your thoughts on the participants today. Please congratulate your favorites in the chat if you like. And why don't we put this poll up? I would like to know, audience, The coolest wow factor science for you today. If I can somehow launch the poll, there it is. You should be able to see it now. What science had the most wow factor for you today? Don't just vote for your favorite or for your class year or for somebody in the same living group as you, please. Vote on the science. What was the coolest wow factor science? We have the results of the audience poll too, but judges, maybe we could go in third place, second place, first place
Yeah,
order.
that was the plan. But I do want to say before, one of the reasons it took us so long is that the talks were all amazingly good. And it was really hard to decide to like eliminate out of these 9 really excellent people, 6 of them, and only give prizes to 3. So like you really should congratulate yourselves, for being able to do this as a, experiment here and do it as well as you have. You all deserve, just really lots of kudos for that. So our third place winner is Juyan Oh. We love the visual. You set up the problem really, really well.
Thank you very much. Thank you.
Yeah. Shall I keep going, Joe?
Feel free to, or if the other judges want to.
You do want to say anything else about what we particularly liked about Julie's presentation?
Well, the visual metaphor was very effective at showing showing not only the tension, but the complexity of the problem.
Did you draw that?
And I got the people doing the tug of war. I downloaded from the web, but everything else, yeah.
Very nice. You've got a very good visual sense.
Thank you very much.
Well done.
Wonderful. Our second place winner is Allegra Hawkins with her Google Map analogy for cancer and spatial transcriptomics.
Thanks.
Again, the visual was terrific, very clearly explained. I think you did a very good job of the presentation, of speaking clearly, and of creating a compelling case for what you're doing.
Thank you so much.
Yeah, and I really thought you explained that well, that connection really well throughout.
And coming in at number 1,
drum roll,
is Maya J.
Yay.
Thank you, guys. Appreciate it a lot.
Great presentation.
I think
passion,
we all
great,
understood. Yeah, we all understood exactly what you were doing, and was really engaging to watch.
Awesome, thanks.
Our work here is done, Joe.
Yeah.
Thank you. Yes, and we'll host breakouts for first, second, and third place. I've posted those links in the chat, and I'll post them there again. But we do have one more award. This was the crowd favorite for science, strictly science, less on communications. And I'll share that if that works. But it was also to Maya J. So Maya, congratulations again.
Thank you,
The
everyone.
audience and the judges agree every now and then on both communication and science.
Son
well
of a
done.
gun.
It always happens that way.
Vivian, Bob Pryor, Holden, Thorp, and Ari Daniel, thanks so much for being such great judges. This is our pilot virtual slam, and Vivian, a great partner for the past month putting this together. And to all of our 9 competitors who volunteered to be critiqued in front of their peers. It's never easy for the betterment of science. We encourage them and you to join us in another slam, virtual or real time. Once again, an archived edited version of this will be available on the MIT Alumni Association YouTube channel within a week's time when we clean it up. Please join the winners in breakout rooms now if you'd like to chat further with them. We'll also keep this main chat open, but we'll go off of video for anybody who wants to stick around and just chat with one another in there in text chat. Vivian, any closing thoughts?
Yeah, it was just super fun to hear what our biology alumni are doing. I'm so happy we've had the chance to do this. I hope we will get future chances to connect with our department alumni in virtual and in-person events in the future. It's just wonderful to see how you're doing.
Superb talks, everybody. Congratulations to you all.
Thank you all, and stay safe and enjoy the rest
Thank
of the evening.
you.
Thank you.
Thank you.
Thanks, everyone.
Stay safe.
Thanks, everyone.
Bye.
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 Thank you.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.